Hello everyone. Today, we will use longitudinal evidence to elucidate the distinct patterns of structural and functional impairments in PCN and DMN-related regions, aiming to reveal the cascade of network dysregulation across the AD continuum. The findings of this study have been published in the journal Med Research. Now, let us formally proceed to today's main topic. Alzheimer's disease is clinically characterized by progressive cognitive decline, neuropsychiatric disturbances, and deterioration in social functioning and activities of daily living. During the progression of AD, the precuneus serves as one of the most densely connected hub regions in the human brain. As shown in the initial 7-network model, see figure A on the left, the precuneus is intrinsically organized into two functionally related brain networks, the default mode network and the central executive network. When the human cortical networks are further subdivided into 17 distinct networks, see figure B on the left, it becomes evident that one of them remains the well-known DMN. The other network was recently proposed by Nicholas, termed the para-cingulate network. The PCN is a para-cingulate subnetwork of the CEN, connected to the cingulate cortex and the core regions of the DMN. It represents a highly correlated, task-active network entirely situated within the precuneus. The figure on the right illustrates the 7 specific regions encompassed by the PCN, whose activations play a crucial role in working memory storage, retrieval, and cognitive updating. As a network closely associated with the precuneus, the PCN may play a crucial role in cognitive processes. However, its pathological alterations during the progression of AD remain largely unknown. Although the pivotal role of the precuneus in AD development has been relatively well established, little is known about how dysfunction within the associated PCN and its interactions with DMN contribute to the pathophysiological mechanisms of AD. Based on the above, we included neurobehavioral and neuroimaging data from 153 participants in the AD&I cohort, encompassing individuals with SMC, EMCI, LMCI, and AD. Our aim was to investigate alterations in the two precuneus-related functional networks across the course of AD and to examine the temporal effects underlying these stage-dependent changes, thereby further elucidating the neurodegenerative mechanisms underlying the transition from the preclinical to the dementia stage of AD. This is a comprehensive graphical summary of the proposed work. After incorporating structural and functional imaging, as well as behavioral data from the 5 participant groups, we will conduct extensive analyses including VBM, functional connectivity, correlation, and temporal effects. By comparing behavioral data, significant differences were observed among the 5 groups in MMSE, CDR, and FAQ scores. Correlation analyses revealed that over time, MMSE scores declined in the AD group, while FAQ scores increased over time in the LMCI, EMCI, and SMC groups. In the VBM analysis, we found that GMV reductions in PCN-related regions initially manifested in the AD group. Over the course of the 2-year follow-up, aberrant GMV in PCN-associated regions also gradually emerged in the LMCI group. Across the 2-year period, the number of DMN-related regions exhibiting GMV decline progressively increased along the continuum from SMC and EMCI to LMCI and AD groups. In the precuneus-centered functional connectivity analysis of PCN-related regions, no abnormal connectivity between the PCN and the precuneus was observed at baseline. After 2 years, the PCN-affected area BA7 exhibited a marked decline in functional connectivity in the AD and LMCI groups compared with healthy controls. Regarding the DMN, at baseline we observed that hippocampal FC in the AD and LMCI groups was significantly lower than in the other 3 groups. After 2 years, we found that FC between the precuneus and the MTG and TPJ in the AD group was markedly higher than in the remaining 4 groups. Bivariate correlation analyses revealed that FC within PCN BA7 and DMN regions, including the hippocampus, MTG, and TPJ, was significantly associated with behavioral measures. A targeted second-level analysis identified correlations between GMV in regions showing group differences and neurobehavioral measures, primarily including the hippocampus, MTG, PMC, BMPFC, DMPFC, and TPJ within the DMN, as well as BA7, BA23, POS, and RSC within the PCN. Further details on these correlations can be found in the manuscript. This study provides novel insights into the pathophysiological basis of functional network dysfunction within 2 precuneus-related networks across the AD continuum. For the first time, we found that gray matter volume and functional connectivity abnormalities in PCN-related regions are associated with global cognition, dementia severity, and functional impairments in both AD and LMCI. Furthermore, structural and functional alterations in PCN regions exhibit age dependence, emerging earliest in AD and progressively affecting LMCI over time. Our findings indicate that abnormalities in the structure and function of PCN and DMN-related regions intensify with the severity of AD spectrum disorders, reflecting the gradual progression from SMC to AD. In summary, this study contributes to the development of more effective therapeutic strategies and diagnostic tools for AD and its preclinical stages.